Many drugs attack cancer by starving tumors of the fuels they need to grow and spread. The basic idea is that if a tumor cell depends on cupcakes to thrive, then take away the cupcakes. But what if cancer craves cupcakes so much that too many cupcakes could do it harm? Could we force feed cancer so many cupcakes that it chokes to death on the excess? That's a strategy Dr. Lucas Sullivan, a Fred Hutch researcher who studies what cancer eats, explores in a recent study published in Nature Metabolism. Writing for Fred Hutch News, I learn something every day about how life works, what happens when it goes awry, and what we can do about it. Come with me to learn something new today, in the lab. So for this study, you focus on cancers that are driven by a specific mutation that helps tumor cells resist drug therapy. And these mutants eat a lot of a nutrient called cysteine, but it was unknown exactly what they're doing with all this cysteine they're gobbling up. When you track down all those uses of cysteine in the cell, you found something unexpected that suggested this craving for cysteine could actually be harmful to a tumor cell.
These cancers have a specific mutation that increases the uptake of this nutrient cysteine. And our current understanding couldn't identify where it all was going. And so we developed a platform to trace those molecules using a technique called mass spectrometry. And we found a bunch of new molecules. And those molecules derive from a specific feature of cysteine, which is that it's incredibly reactive. And it seems like these cancer cells accumulate cysteine to such high levels that it starts undergoing chemical reactions with other nutrients in the cell. And so normal nutrients that aren't normally exposed to this kind of reaction, it starts soaking them up and generating these new products that seem kind of a specific feature of these cancers, that they have a lot of these new molecules present in them. And so we thought about that and we realized, hey, this sounds like a bad scenario where normal nutrients are being reacted with this cysteine molecule. And so when we looked at these cells, we found that the more cysteine they had, actually the more toxic it was to them when they were growing slower as a consequence.
So this mutation that makes them eat so much cysteine sounds dangerous to them. Why would these cells keep this mutation around if it's so harmful?
It has to be the case that these mutations are promoting cancer. Otherwise, they wouldn't appear, they wouldn't be kept around. But that doesn't mean that every mutation is universally beneficial, right? So this is a stress response pathway. It's an alarm bell. And part of the programming of this pathway is to increase cysteine for drug detoxification, nutrient metabolite detoxification purposes. But that doesn't mean that it's always good, right? And so having an alarm bell on all the time may be net promoting for the cancer, but that doesn't mean that it doesn't have drawbacks as well. And so our research would say that this is a pros and cons situation. And so it isn't always the case that just promoting the whole program is universally good. And you might have these drawbacks like the ones that we found where cysteine accumulation can cause these reactions with normal molecules and can be deleterious to cells. And so we think that might be something we could leverage for therapeutic benefit. And that's an ongoing area of research.
So you tip the scales by feeding it too much cysteine, and that kind of gives you a new way to attack these. You can force feed it.
By modulating the amount of cysteine that's available for them, we can kind of leverage this difference between cancer cells and normal cells, put them into this cysteine stressed condition, and then potentially, leverage that directly or potentially the consequences of that to separate cancer cells from normal cells and develop new therapies.
But if you do this, wouldn't that be also harmful to the healthy cells?
Normal cells are going to be unresponsive to changing the amount of cysteine that's in the environment and the amount that gets into the cells. And the cancer cells are specifically going to be the ones that are unable to control the amount that gets in. At the end of the day, cancer cells are derived from us. They're very similar to us. And so we're always looking for ways that we can separate normal cells from cancer cells based on the specific features of those cancer cells. And so one of the goals of, say, precision oncology is to find those specific differences. And if this unrestrained cysteine uptake is one of those opportunities, then we want to learn more about it and then build off that for future therapies.
So that ability to differentiate between the cancer cell we want to attack and the healthy cells we want to preserve, that's kind of the idea of what we call precision oncology, right?
Yeah.
And I think that sometimes people are thrown by that because it implies that the oncology we did before wasn't so precise, and now we're going to be precise.
All cancer treatments are ways of differentiating normal cells from cancer cells. And the origins of oncology research and precision oncology is to take advantage of those differences. So one of them is that cancer cells grow faster than normal cells. They proliferate faster. And so the conventional chemotherapies have been things that specifically target dividing cells. Now, lots of cells in your body are dividing. And so as we move towards the future, our goal is always to find more and more nuanced and specific differences between cancer cells and normal cells. And so as a cancer metabolism lab, we are thinking about that in terms of how's the metabolism of cancer cells different from normal cells. And in this case, they have a very specific cysteine-related metabolic difference. And so we think by further understanding that, we can develop new therapeutic agents that specifically target this precise difference between cancer cells and normal cells.
This is John Higgins, reporting for Fred Hutch News.